and looking at what's going to happen with this formation of urine. That, of course, if you remember, is done in stages. And as we talk about urine formation, the kidneys as a whole, excretion of wastes, okay, the first stage is, of course, that formation of the filtrate. Now, where is that filtrate formed?
Where is it?
The glomerulus into the glomerular capsule. Okay, so stage one would be the forming of that filtrate. Now remember, at this point, the proper term is filtrate, because that's kind of what's happened. We've had the blood flow into that glomerulus. We've had those materials kind of get moved out into that glomerular capsule. It's a combination of a lot of things. We've got water, we've got salts, we've got sugars, we've got a lot of materials in that filtrate. So now, once we have the filtrate, we're going to begin the second stage. Because once we have the filtrate in the glomerular capsule, we now encounter that portion of this nephron, and don't forget that that nephron is the functional unit of the kidney. So, in that nephron, where I've got that glomerulus, all right, the first thing I'm going to encounter, and y'all know that's not structure, okay, but the first thing I encounter, remember our proximal and distal convoluted tubules, okay? So the first one I'm going to hit is my PCT, okay? Is that right? Proximal convoluted tubule. Don't forget that we have our structure that plays an important part. And a lot of this is that simple squamous, simple cuboidal, okay? And don't forget that this is a tubular structure, meaning there's a lumen, and that fluid is flowing through that tube. And because of the cells that are present and their structure is how we're going to have the ability to move material. So we have those two types of nephrons, and one of them, the vascular structure that they're in contact with, all those blood vessels, is like around this glomerulus, the PCT, and then what we're going to eventually hit as our DCT, okay? And then remember how we're going to see quite a few nephrons entering that collecting duct, okay? So I have my collecting duct, get to my little renal, the papilla area, into a little calyx, which kind of goes into bigger calyx, if I remember that right. Okay, so in the part two, in stage two, we're going to have this termed tubular reabsorption and secretion. Now here's what's going to happen. In one of those types of nephrons, where I've got the capillaries all around my glomerulus, my PCT, my DCT, and then in another one, as I entered the loop, okay, if you remember, I had some vessels that kind of did this number, okay? I had my cortical and my juxtamedullary, all right? So, as the blood enters into this glomerulus and makes its way through, and materials become my filtrate, we are in, do you remember how many, what percentage of nephrons had the structure of the capillaries around this portion, my cortical? 85% had their capillaries around the cortical portion. So, as I enter my PCT and I'm in close contact with these capillaries, that filtrate that contained water and sugar and salts and all this other type of stuff, that's stuff that I do not want to continue forward as waste. I want it to make its way back to the bloodstream. So I want it to get moved so that it can encounter those capillaries and go back to the bloodstream. Is everybody following me? Okay? So that is reabsorption. Because I've absorbed it once, it became filtrate, and I'm going to send it back. So my bloodstream gets to reabsorb it. Now, the secretion portion is saying that along these vessels that we're finding that are encountering other parts of the tubes, if there's something still left that doesn't need to stay in the blood, the bloodstream, these vessels, it can actually move back out into my filtrate. So my blood vessels secrete it back to the filtrate. Does that make sense? So that's why we have those two terms. We have the terms of reabsorption and secretion. So what we're going to do during the next stages, we need to make sure that that filtrate becomes urine. We're going to have to get those chemicals removed from filtrate or from blood back to filtrate. And the formation from this point goes through the tubular portion of my proximal all the way to my distal convoluted tubule. So I'm looking at now what's going to happen from this point to this point before it enters the collecting duct. Okay? So the PCT alone, that proximal convoluted tube, reabsorbs, meaning it's going to, that's almost 65% of that filtrate material is going to go back to the bloodstream. But now let's go back to thinking about our amount of fluid that's flowing through this system per day. We said, what, about 180 liters per day? We'll just go with that number, okay? Pretty sure I'm not going to be able to keep up with the math correctly, so y'all just bear with me, okay? All right, found out yesterday I'm not good at math, but we're going to keep it at that, okay? So, 180 liters. 35% after the PCT is still going to remain, because we definitely don't want to urinate out that much, right? So 35%, y'all help me here. 18, 18, 18. 54 plus 9, 63. I'm still at 63 liters of fluid at the end of that PCT. Do I want to urinate out 63 liters? Me personally, no. Okay, just saying. Now, that means for the rest of that tubular structure, I need to depend on that cellular structure that's present to move these materials. So, for this reabsorption portion, where I'm going to get materials returned back to the blood. Remember, water, salts, glucoses, it's got a lot of stuff, okay? So, this is my structure that's present. I have my simple cuboidal on that tubular portion. It also has the microvilli. This would be what would be my lumen, the middle portion of that tube. This would be where my filtrate is passing over. So the filtrate that got made in this glomerular capsule, that fluid is now passing through those tubes, basically flowing across. These cells are here and that fluid's here. Okay? Because of their structure, they are really good at allowing materials to move. Materials will get to move either between the cells or across the cells. This would be my basal side. This is my apical side. And remember, in 85% of those nephrons, they're in close relationship to a blood vessel, which is simple squamous, also made for movement of materials. Is everybody following me? Okay, so here's what will happen with the PCT. Like I said, we can have movement of materials either through the cells or between the cells, across the cells or between the cells. In some cases, we have what's called solvent drag, meaning if water or a substance is moving in one direction, one will follow the other. Now remember, at this point, a huge amount is water because it's the fluid that has made its way, because it's our blood, and blood is a fluid that we're very dependent on with water being present. So in my PCT, look at what is going to get to happen to materials that are in my filtrate. In my filtrate, I've got sodium, I've got glucose, I've got H pluses, I've got chlorines, I have sugars, I have magnesiums, calciums, phosphates, urea, uric acid, blah, blah, blah. I got a ton of stuff in that filtrate. Now, the way that some of the materials are going to move. Now, note, one of the things that happens, I need ATP, okay? The kidneys use a huge amount of energy to carry out their processes. So I'm going to need ATP to make energy. I need my sodium-potassium pumps to work, okay? But look what happens with glucose. Glucose that's in the filtrate at this point is where it moves. It is at this point of the nephron structure that the glucose moves. If the glucose makes it past this point, chances are it's going to end up in the urine, and therefore end up being